Hope everyone can see it. So yeah, thanks, Andrew. So I'm just going to focus on how we developed a spin-out company based on an anti-cancer drug that we developed in University of Wolverhampton. So the group that developed this work is Experimental Cancer Therapeutics Group, led by Professor Wong, and I'm a group member, and I eventually moved to the spin-out company, and also looking after something in the university at the moment as well. So I'm not going to tell too much details about the background. The purpose is just to give a quick introduction within the given 5-6 minutes time. So if you have any questions or anything, you can contact me, or you can visit our website, disulfican.com. So the healthcare need that we would address is oncology. Basically, we are trying to understand why, despite having so many drugs in the market, why cancer become resistant to drugs, and why cancer spreads to different places. And this is the reason why 95% of cancer patients eventually die. So we want to address this area and develop new drugs in this area, which majority of bigger universities and wider world of cancer community is focusing as well at the moment. So one of the key understanding that we have is when the tumor develops rapidly, there is a low oxygen core that is developing inside the tumor, and it drives a series of molecular changes in these cancer cells that gives them the ability to become a cancer stem cell phenotypes, we would say, because these are cells that are reversible from a fully developed state to a stem cell state. Stem cells are usually early stage cells, and these cells are extremely resistant to drugs. So when you give a chemotherapy, you can comfortably kill the bulk of the tumor, but you won't be able to get rid of these cancer stem cells. And because they have the stem cell phenotypes, they have the ability to come back or bring back the entire of the tumor back in place, which is the reason for relapse. Also, some of these cells gain the ability to move from one place to another, and that becomes more metastatic and then spread to different organs, which eventually becomes untreatable with any sort of drugs that are available. So we think that we need new drugs to target this cancer stem cell, and with this in mind, we wanted to search for new drugs. But drug development is an expensive process. It also takes close to 15-20 years to develop a drug and put in the market. So there is a global trend now for using an existing drug and then quickly translating. Common example I would give is Celgene pharmaceutical company, a global giant, which took the 1950s drug thalidomide, and then now they have 3 different anti-cancer drugs for multiple myeloma. And recently, that company has been sold to Bristol Myers Squibb for about $79 billion. Johnson & Johnson also have a stream of repurposed drugs. And also now, we have seen from COVID situation, people trying to use hydroxychloroquine and chloroquine, this kind of anti-malarial drug or antiviral drugs like remdesivir or dexamethasone, which is a steroid, coming to COVID Act. So this is the example for repurposing. Like that, we have taken an anti-alcoholism drug that has been used in clinic for more than 60 years and has been used on millions of patients at the moment. So we have taken that drug and we have demonstrated the anti-cancer activity. And we have specifically pointed out that disulfiram has a structure which has the 2 diethyl dithiocarbamate molecule joined together by a thiol group. And this thiol group is highly reactive and is important for the anti-cancer activity because the reactive thiol group is able to chelate metal ions like copper or zinc and instantly generate reactive oxygen species that kills the cancer cells. So we also hypothesize cancer cells have an extremely high amount of intracellular copper because of their fast growth and metabolic needs. They need a lot of protein that needs copper. So if we deliver disulfiram, and then they will be able to transport disulfiram inside and then combine with copper or bind with the orally administered copper outside and kill the cancer cells. So by doing this, we can selectively kill cancer cells and then spare normal cells, because side effects is the most important problem with a new range of drugs that are being coming to the clinic at the moment. So we have demonstrated disulfiram's activity in wide range of cancers. In our group, we work with about 8 to 10 different type of cancers already. But when you want to take this to commercial, then we want to focus on only a couple of them. Another important finding from our group is that disulfiram is a commercially available drug, and the available version is an oral version at the moment, which when you put in the liver, it's going to split into 2 DDC molecules. But unfortunately, in the liver, this thiol group, reactive thiol group, is metabolized and then methylated and blocked completely. So the binding with copper never happens, and the cancer killing ability is completely blocked. And the short half-life is another big problem for disulfiram, because the oral disulfiram is the only version at the moment, unfortunately. So that 17 clinical trials which have been using copper and disulfiram for cancer treatment have failed already, and we found that this is the reason behind the discrepancy, why disulfiram is not working in clinic but works very well in the laboratory condition for cancer. So our group has been researching this for about 10 years, and then we developed a PLGA encapsulated disulfiram, because our hypothesis is if we were able to block this methylation and bypass the liver and deliver intact disulfiram next to the tumor site, we will be able to achieve its anti-cancer activity. So we developed an encapsulated version, and we were able to extend the half-life for 7 hours from 4 minutes. And then this is a slow release and increased stability, disulfiram. And this is very suitable for injecting locally. So there are specific cancers which need drugs that can be injected locally. One example is mesothelioma, which is an important cancer caused by asbestos, and the numbers keep on increasing globally at the moment. And there are no effective drugs in the market. Another example is glioblastoma, which is a brain tumor, which many of us already know how serious brain tumors are. So this is our finding. We tried different range of encapsulation, and we have patented a couple of them already, and PLGA is one of the prime products that we were able to successfully use. And like I said, for commercial focus, we have to use only one cancer or two, maybe, because that's what companies intend to do. So we have chosen malignant mesothelioma, and the global incidence rate is, although less than 1%, it is estimated 750 million people are already exposed to asbestos, and it will develop in the next 20-30 years. And what we go with in this scenario is just 2 drugs that are commercially available at the moment, that gives less than 10 months for the patient to survive, because most of the patients either relapse or get resistance. So that's why we focused on this drug. And these drugs are, this disease, and this disease is categorized as orphan disease because of lack of effective treatment at the moment. So we want to take the spin-out company strategy to an orphan drug disease indication strategy. So we have developed an initial project to have a proof of concept. So we scaled up the disulfiram microparticles in an industrial setting. We've already produced GMP quality particles. We have tested the efficacy in a GLP setting. Basically, we have to do it in a contract researcher setting approved by MHRA, and we have completed those proof of concept. Now the company is trying to move into orphan drug disease route because of the 8 years of market exclusivity that we will get.
Sorry to interrupt, Vinodh. I was going to say, we've come to the, if you can wrap up now, we've come to the end of the,
so the end is now the company is focused on developing the next stage to move to clinical trials in 2022. And if we were able to successfully translate for mesothelioma, we can translate for other cancers which are highly aggressive and has no treatment as well. Thanks.